Hi everyone, today we'll be talking about blood and the aging of blood, blood donations, the aging of hematopoietic stem cells. So let's start the ride. The whole project started with this article. It's an article about the analysis of an old lady's blood. She was 115 years old when she died, and her blood was analyzed, and they discovered that only 2 hematopoietic stem cells were responsible for her whole blood system. Well, a hematopoietic stem cell is a blood stem cell. It means the same thing. A stem cell has 2 specific properties. One, it's capable of renewing itself without aging. So it renews itself, it can multiply and make daughter cells that are exactly identical to itself. And this theoretically could continue forever and ever. And the other characteristic for a stem cell is that it's capable of specializing and engaging itself into different cell lineages and become a specific cell, like a red blood cell or a white blood cell, and so on. This started a discussion between some friends and me about what this discovery implied. What we were worried about was that blood donations would use up your blood system and make it age earlier. And so if at the end of your life, even if it's 115 years, you only have 2 blood stem cells left, if you've given your blood before, then maybe you have none. This inspired a friend of ours to draw this picture. He's a very talented graphics designer, and you should check out his website. So you see person A is someone who doesn't give his blood, and at different periods of his life, maybe he has, I don't know, iron deficiency or something that makes him lose a bit of his blood capital. And person B would be a blood donor, and he would do multiple blood donations throughout his life. The green person, it would be from injuries that he lost blood. So maybe twice in a lifetime is a little bit too much, but. And so throughout your life, your capital, your blood stem cell capacity would go slowly downwards, and your body needs would go up depending on illness and different kinds of pathologies. And so, as you can see, the hypothesis was that person A would live longer than person B because person B would reach the limit of the body needs earlier. Another friend illustrated the theory with 2 questions. She wondered if each time you give your blood, since there are circulating blood stem cells in the peripheral blood, whether you could lose them with the blood donation. And the second thing she worried about was whether or not, if you give your blood, giving your blood makes you lose blood. It's kind of the same thing. And that means your body has to make the blood again. It has to renew the blood. And that would mean that the blood stem cells would be stimulated to renew the blood. And would that be significant? So, do we lose blood stem cells when we give blood? I had a hard time finding information about this question. I finally found this study that's from 1994, which is quite a long time ago. It was a study comparing different protocols to mobilize the CD34 plus cells. CD34 is a blood surface marker that you can use to identify blood stem cells, but it's not quite perfect because it's not specific of blood stem cells. It also stays there with blood cells that are a bit more engaged into different pathways. So it doesn't only give you the blood stem cells, it also gives you other cells. And so in this study, they compared different protocols, and they compared the protocols to a steady state normal person, well, to 5 actually. And so this is what they obtained as a result. They obtained this was the percentage of cells of CD34 plus cells that they found in the nucleated cells that they isolated in these people. And I realized I should have put also the bone marrow steady state count, because that would show you that it's 100 to several hundredfold more in the bone marrow, and that these numbers are not that big. But still, that does mean that when you give your blood, you will lose some stem cells. The other question was if the blood donation would be a significant stimulation of the hematopoietic stem cell division. So if giving your blood would force your system, your blood system, to multiply a lot, and that way would make it use its potential up. So my question, maybe you're not going to see the link right away. My thought was, if you give your blood and the stimulation of the stem cells is significant, then that means the stem cells will divide more. If they divide more, they have more chances of making errors while they divide and having mutations, and mutations lead to cancer. So I thought the easiest way to see if the stimulation of the blood system was significant was to check if there was more cancer in the blood donors. So I found this study and several others which said that the risk of cancer among blood donors was not superior to standard population. I don't know how you should say that. Standard population sounds weird. So if the cancer risk is not higher, then that means to me that probably the stimulation of the blood cells is not significant. But it's an approximation, and I don't think we can conclude with only that. Unfortunately, I couldn't find other ideas. All these thoughts made me wonder what this hematopoietic stem cell aging was. How do the blood stem cells age, and what does that mean? I found articles, lots of articles, several, unfortunately, that were not free of rights, so I couldn't read them entirely. I could only get the abstract from PubMed. So the first one is talks about the differences between young and old blood stem cells, sorry. The second one talks about what we see in the aging hematopoietic system. Don't hesitate to pause the video and read these abstracts. They're very interesting, although short. And then I found interesting articles, several of them again, about telomeres in the hematopoietic stem cells. Telomeres are the end of the chromosomes. They are very short sequences of DNA that are repeated many, many times. And those telomeres, at each cell division, there's a little bit that's lost because the enzyme, the DNA polymerase, that copies the DNA, cannot reach to the end of the DNA. And that means with each cell division, there's a little bit of the DNA that gets lost in those telomeres. At some point in regular cells, we discovered that the erosion of the telomeres was too important. So the loss of these DNA sequences was too important for the cell to continue living, and it started a process to destroy itself, to kill itself. So if you remember what I was saying at the beginning of the video, stem cells have two properties. The one I'm referring to, is that they have an unlimited capacity to divide themselves and stay exactly the same. That's what we thought. Because this article and others, it's the most recent I could find, but it's not the only one, tells us that the telomeres in the blood stem cells are not spared by the aging process. So, like other stem cells, they have an enzyme complex named telomerase that works. It's active in those cells, in all the stem cells, but they saw that its expression was limited in the hematopoietic stem cells. This telomerase complex, I have to tell you a few words about it. It's kind of funny because I was telling you that the telomeres get eroded. They get smaller and smaller with each cell division, and this telomerase complex, it has the function to lengthen them again. I won't enter the details because it's complicated without drawings. But maybe some other time I'll tell you about it. The second article here is, well, I couldn't read that one entirely either. I could only have the abstracts, just the way you have it here. So I don't really know what their conclusions were, but apparently there are microenvironmental effects that play a role in the aging process of the blood stem cells. Another paper, and this one I think will be my dad's favorite because it's full of equations and complex formulas. It was also about blood stem cell aging, and its point of view about the aging process was that the lifespan of the cell was limited by the repair capacity of the cell. So this is an interesting illustration they put in their article. So they took one blood stem cell and they transferred it to, they were working on mice, as most of the time they are with blood. So they used one unique stem cell, they transfused it, well, they grafted it, sorry, to an irradiated mouse, so a mouse with no blood marrow at all, nothing. So no blood cells, no stem cells, nothing. So they used this one cell, they grafted it to this irradiated mouse, sorry. And so they saw that there was first an expansion of the blood stem cells, but it stayed the same because, as I was telling you, it multiplies by copying itself exactly. And then there was the lineage expansion, which means that the stem cell would create cells that would engage into different lineages of the blood system, as red blood cells, white blood cells, cells that make platelets. And at one point, they would exhaust their potential, these cells, and die. But what was interesting was that all daughter cells became extinct at the same time. This suggested that the lifespan is programmed and transferred from one cell to its next generation. I'm not going to go in depth about epigenetics because it's very complicated. It's globally, it's everything that's around the DNA and that changes its expression. So, for instance, you have methylation. I'm not sure that's the word in English. I hope so. So we have methylation of different parts of the DNA, which makes its expression higher or lower depending on the degree of methylation. And this article told us that there was an epigenetic regulation involved in the aging of the blood stem cells. So many different ways of aging, many different ways, many different things discovered, and Very interesting, different paths. Um, After all this, I was worried about the lifespan of a red of blood stem cell. Sorry, because with the last article we just saw, it seemed like one blood stem cell would have a limited lifespan and then nothing left. I found some answers in very old papers. The one on the left is from 1990, and the tiny bit on the uh, down part of the screen is from 1965. So the 1990 paper referred to a theory that was exposed in the 1965 paper, but I couldn't read the 1965 paper, so I copied an extract of the 1990 paper. Their idea was that once you notice that a specific stem cell only lives so long, well, how could one human being live his whole life with stem cells continually renewing themselves if the stem cells die at some point? That would mean that at some point you have no stem cells left and then no blood. So their theory was that only a portion of the total stem cell pool would be active at a given time. Hematopoiesis would be maintained by only a succession of short-lived clones. Clones would be one cell. Oh, sorry, I'm going to go back. So this is a clone. You have one cell, it divides into multiple daughter cells that are exactly the same. This is a clone. So at one point, only some of the stem cells would be active, and then they would die, and other stem cells would take the next part of the blood system generation. And so that was an interesting hypothesis. What they say is that when they look at the mutations in the DNA, they see that for extended periods of time, some of them have no changes. So that should mean that they're not very active and not multiplying very fast, not dividing, sorry, very fast. And they have other populations of blood stem cells that do change and have mutations. So that would mean that some of the cells are active at one point and others are not. They also provided evidence that the stem cell population can clonally expand during the regeneration of a new hematopoietic system. It was mostly the same thing as the one with the picture with the little round blue dots. This study is also an old study. It's from 1973, so quite a long time ago. It studies the lifespan of the blood stem cells in mice. It studies it in mice because of their quick multiplication rate, so it's easier to study mice than to study, I don't know, human beings, for instance. The study is as follows. You take one initial mouse and you want to determine how long her stem cells are going to work and are going to be able to divide and produce a whole blood system by themselves. To check that out, you have to use irradiated mice, and from the first one, the normal one, you take blood stem cells from her bone marrow and you graft them into an irradiated mouse, and those stem cells are going to develop, I don't know, and make the blood system again whole. Then you take a third mouse and you irradiate her, and you take the blood stem cells from the second mouse, so already renewed from the graft, and you transfer them to the third mouse, and so on. They saw that from one mouse you could use the stem cells for 73 months. Actually, a mouse lives 1.5 to 2 years, And so 73 months is about 6 years, so it would mean 3 to 4 generations of mice, which is not bad for one stem cell. So apparently, at least for mice, because I couldn't find information about human beings, obviously you wouldn't do the same study. So with mice, at least, the stem cells live a lot longer than the mouse's lifespan. Now let's go back to the first article, the one that started everything. I checked the article itself out because I wanted to know what they were really demonstrating in there. So what they say is that they couldn't check the blood stem cells and their telomeres, but they could check the white blood cells and their telomeres. And obviously, if the stem cells have short telomeres, then the white blood cells emerging from those stem cells will have small telomeres too. So as they see that there are small telomeres in the white blood cells, they suppose that the telomeres are short too in the blood stem cells. And they conclude that probably the aging of the blood stem cells and their lifespan is determined by the shortening of the telomeres, and not by mutations that would, I don't know, inactivate enzymes or stuff like that. So to sum up, we have stem cells circulating in our blood. We have blood donations that might stimulate the multiplication and division of hematopoietic stem cells. And we have aging of those stem cells that might limit their lifespan, even though in mice we've seen that the mice can outlive 3 generations. The blood stem cells can outlive 3 generations of mice. So all this would make us wonder, why would we give blood when we have this risk of limiting our lifespan? And I have 2 answers to that. One is the one that's down there for those of you who can read French. It's one friend who was saying that, yeah, maybe it would shorten your lifespan, but it would do that so little. And we all agreed that apparently it was very little, otherwise we would have seen that already from the frequent blood donors. So the shortening of your lifespan would be very little. And well, giving your blood is a very noble activity, and it helps you feel accomplished, and you save other people's lives. And everyone knows that people who are happy and feel fine with themselves have better lives. So that's one reason to give your blood. And the second one is my idea. It's that since, well, the lady that was explored was 115 years old, and she still had 2 stem cells left, which is not very much. Well, she still had those 2 stem cells, so maybe she could have lived, I don't know, 5, 10, 15 years more. So maybe the limit of our lifespan would be about 130 years, but now we're so caught up with other kinds of disease that the shortening of our lifespan, I don't think, is very significant yet. Let me phrase that another way. We could, hypothetically, if the stem cell factor is the limiting one, we could live until 130 years, but no one actually currently lives until that time, because there are so many other diseases to take care of. So before it's going to be a limiting factor to a human lifespan, I think a lot of time is going to pass. I picked a couple of articles for those of you who want to think about this subject a little bit more. So number one is kind of yucky, so if you're sensitive, please don't listen to me. Scientists found that blood from young mice rejuvenated the muscles and brains of old mice. What they did was parabiosis. I don't know if you pronounce it that way, but it's, to simplify, you open the side of one mouse, you open the side of another mouse, one is old, one is young, you fuse them together, you stitch them together, and so the blood circulation is, the both blood systems develop together, and they communicate so that the young the young mouse's blood goes into the old mouse, and the old mouse's blood, well, all of that mixes up. And so with this kind of study, this is one article from this team's studies, but it's not the only one. It's the first one, the one where they did this experiment, and so they saw that the blood from the young mice would rejuvenate the muscles and brains of the old mice, and afterwards they identified the GDF11 factor, which is a protein, I think, and when they gave that to old mice, they had the same effect on rejuvenation of muscles and brain. So they think it might be a very important factor in aging, in the aging process. Maybe if you don't have it anymore, then you age faster, or, well, I don't know, maybe while you age, you produce it less and less. Or so if you want to think about that a bit and talk about it, don't hesitate. And the other one is another interesting thing. We were talking during our discussion with my friends about the fact that tracking hematopoietic stem cells was very difficult. That's what I was telling you at some point, that you can use the CD34 plus characteristic, and you can use that, but it's not enough to let you distinguish hematopoietic stem cells from cells that are a little bit older. Here I put the two ways I knew of to track stem cells. One is using antibodies and fluorescent dyes, and the other one is kind of like what I was telling you with the mice earlier. When you irradiate mice and you put the stem cells into the second mouse, you use mice, and if the cell you put in the second mouse can renew the whole blood system, then you can tell that the cell you used was a blood stem cell. But you can only be sure once the system has been renewed, and you can't pick the unique first stem cell anymore. So it's a limited technique. And so this article talks about a new technique that uses 3 different technologies, and it's kind of interesting. So if you want to check that out too, yeah, you can do that. So here we're at the end of our journey. I hope you enjoyed it. I hope it made you think and ask yourself questions. Don't hesitate to comment down here and to check out the different articles. I'll try to put a link for all of them. If you have other interesting articles, I'm interested too. If you have other subjects you want me to discuss, well, don't hesitate to send me an email at medicalnews33@gmail.com. Thank you.